Thanks very much. It's really an honor and pleasure to be here. This number 50 years is so huge, and I really love that this symposium is positioned between looking back into the first 50 years and really looking forward into what can and should be done in the times to come. I will start with a lesson that we got from Hippocrates, the father of medicine. He was the one who realized that cancer is a systemic disease, and he proposed that curing cancer can only be achieved if we take I'm sorry. Okay. So he proposed that curing cancer can only be possible if we take a systemic approach, if we consider the whole body instead of just eradicating the primary tumor. It's also interesting that he gave the name to the field. So what you see here are actually these finger-like projections of the evading cells, and it reminded him on a crab or cancer, and this is actually how the disease is being called today. Cancer is an incredibly complex disease. So we all know that it involves interactions of multiple cells, extracellular matrix, gradients of oxygen, many bioactive molecules, physical forces, immune cells, etc. Cancer is also an incredibly clever disease because cancers diversify, they evolve, they adapt in order to evade the ability of the body to actually fight them and to eliminate them. And finally, most cancers change in a very radical way from one individual to another. So when you put all this together, it becomes very clear why the field is pursuing this approach, which is resulting over the last few decades from the convergence of many different disciplines to really have this comprehensive approach to the problem, and why we are trying more and more to develop patient-specific models of cancer. So what we see in vivo in the patients, we're really trying to recapitulate in vitro using a number of different bioengineering and scientific tools. I will try to show you today that engineering tiny human tissues can be a methodology that can support cancer research. The tissues that we are making are very small in size, they look very simple, but actually they do have a capability of emulating, of recapitulating some of the organ functions. I will show you an example here. So what you see in this slide is a model of human leukemia on a chip that we start building by starting from a mineralized matrix that we populate by bone-forming cells, stromal cells, and vascular cells in order to form living human bone marrow. This takes about a month. After that point, we introduce into this construct where you have cells, different cell populations organized the right way and interacting with matrix, we introduce hematopoietic cells. All these cell populations are derived from the same source. You start from the very same induced pluripotent stem cells. So these are the cells you get from a very small blood sample. So it's genetically cleaned, it's patient-specific. After a period of time, you get a model of functional bone marrow where you can now introduce the patient's leukemic cells that are not processed, that are just harvested from the patient. And as you see here at the right, you end up forming a module of the human leukemia. Again, the module that has many helpful readouts and that is patient specific. So it's an individualized approach. The reality is, as we all know, that most cancer deaths are due to metastasis, not to the primary tumor. So metastatic cancer is this super villain with superpowers to really evade any ability of the organism to fight this cancer. Metastasis is very complex. It involves many different steps, and then this simple cartoon here is just reminding us that it starts from the primary tumor. The tumor is shedding cancer cells, not all tumors, but some tumors do. These cells find their way through the endothelium of our vasculature, get into the vascular circulation, and then from there, they may go back through the endothelium to target one or more distant organs. It can be brain, it can be lung, it can be liver, maybe one or more of these organs. So it's very clear that modeling metastasis, building a model of metastasis, would really require having a combination of primary cancer cells, immune cells that are very, very important because of their interactions of the cancer cells and also with other organs. You need target organs that may be become the sites of metastasis, and you need vascular circulation. So this is now like a much more complex problem than what I showed you with like one single organ that we use in conjunction with the cancer cells to make a model. So this brings us to organs on chip. What you see here is actually a model that has evolved over the last 10 years or so by converging some of the advances in stem cell biology and tissue engineering in order to build controllable and biologically meaningful models of human physiology and human pathology. So to build the model, you start again from these induced pluripotent stem cells. They can be derived from any individual, and they have this ability to give rise to practically all populations that you can find in our body. So you will get the iPS cells, you will differentiate them into all cell lineages that you need to make the tissues of choice, because each of these organs, liver or heart or brain, will have many, many different cell lineages, not just one cell type. You make these tissues, you mature them in culture so that they can assume the adult-like structural and also functional features, and then you introduce them into these platforms like one tissue type per chamber. The next step is that you link them by vascular flow. So the difficulty in building these models, why did it take 10 years and not 1 year to get where we are now, is that you're really addressing two conflicting requirements. For a tissue to maintain its specificity, its phenotype, as we say, it needs to be kept in its own compartment with its own factors that are optimized for this particular tissue. Our heart and our liver are two very different environments. On the other hand, you want these tissues to talk to each other, to communicate. And the way we address this is actually to try to mimic how body does it. So what our body does is we are alive because of compartments and boundaries. This is the whole secret. So we place each tissue into one compartment. Underneath the tissue is endothelial barrier. Underneath endothelium, there is a vascular flow. So this is what you see here. And then just for reference, you see the size. It's the size of a microscope slide, or when we scale it up, this is really 96 well-plate format. So you can populate this kind of tissue chips, organs on a chip, by many, many different tissues. So here is a menu of options that we have now that is constantly growing. We do have this ability to generate many different small-sized tissues, and we call them organs on a chip because they do have ability to recapitulate organ functions. For example, our liver tissue is recapitulating liver metabolism. Our heart tissue is recapitulating the contractile function of the heart. So it's really beautiful because you can pick and choose. And here are some of these tissues are highlighted that are of particular interest for cancer research, such as bone marrow and liver. They're all also we have metabolism, vascular cells, we have tumors, and we also have the immune cells. These platforms are very easily assembled. They're really plug and play. So you see here at the left in this little video, we first place these endothelial barriers, and then we park one tissue after another into these chambers, bring in culture medium, and when this is done, you connect them and establish perfusion. So these little dots that you see here at the right are actually indicating that what we may have in circulation, and these are cancer cells, immune cells, extracellular vesicles that the cells are releasing, and many bioactive molecules. And this is how these tissues talk to each other. They talk to each other by cells crossing endothelial barrier, and they talk by different factors that they are either releasing or receiving. So it sounds like very simple, and it is simple, but actually it does have ability to model some of the complexities of the cancer. And I'll give you an example of the study that we have been conducting using this model system. So in this particular case, we combined 3 tissues that are known metastatic sites for breast cancer. So this is bone and liver and lung. These are not all metastatic sites, but those tissues are known to be attacked by breast cancer cells. And we added heart as a tissue that is very rarely, if ever, attacked by metastatic progression. So we have these 4 tissues in this platform, and then into the vascular perfusion, we introduced primary breast cancer cells, and we introduced PBMCs, so peripheral blood, essentially immune cells, like the whole menu of immune cells. What we noticed in this system are 2 very interesting things. One is that different types of cells resulted in metastasis. And they would go into bone, liver, and lung, but these cells would not go into the heart. Not in one single experiment we had any invasion in the heart. Even more interestingly, when we used the cells that in the whole organism showed propensity, a preference for bone metastasis, we saw the same thing in the chip. Cells that were going to lung would also go to the lung, to the chip. This is very early, this is very initial. You see these examples of the bone tumor and lung tumor, but this is actually promising in a way that we see the cells being really smart and responding to the right cues that we are giving them. So there are possibilities to really develop different types of models for different types of cancers. To add 2 things, one is, as I mentioned multiple times, this is all can be patient-specific. So we can study cancer 1 patient at a time. And these tissues are very small. The number of cells that you need, cancer cells, immune cells, is very small. So you can get very far, do very many experiments with reasonably small samples that you can get from the patients. So let me now just loop back in closing to some of the opening remarks that we are also talking about next 50 years. And I was thinking, like, where are we going, or like to enable people to do research that may benefit cancer research in the next 50 years. Here at the left, you see the platform as we have it today. This is it looks very pretty, but this is an artisan device. It is made for laboratory research. It requires a lot of training, and our students take time. They just get better and better using the platform. This is not the right setting for disseminating this platform, giving it away, to companies, to clinicians, making a little bit broader impact on the cancer research. So one direction of our research is today standardizing these platforms, making them maintain their many functionalities. They have sensors, they have like a lot of capability to monitor tissue function, but make them as easy to use as bell plates. So we would really like to make them user-friendly so that you don't need super trained bioengineer to operate these platforms. And finally, I think that these kinds of studies, which are really team science, which are being conducted in collaboration with many of our colleagues here at Columbia, many of our collaborators are now also here in this room, we hope that these studies will also help perform another function, which is a function of our research university, which is to train the next generation. So we are preparing people today who will be going out of graduate school and working over this next 50 years. So I would just like to close with this and really thank you for inviting me, and thank you for organizing this really incredible celebration of the 50 years of the Cancer Center. Thanks very much.